Now, the final technique that I want to just quickly look at is something called DNA hybridization. Now, you don't need to be able to recall how to do this technique, but you may come across exam questions where they ask you to interpret the results of the investigation and use that result to determine the relationships between organisms. Okay, and it's basically using the same principle of working along the lines of working out how much DNA they have in common without fully working out the base sequence of the DNA strands. And what this technique's based on is the fact that DNA, we know DNA has a double helix structure and it will separate into two strands when we heat it up. Now, when it then cools down again, you then get reforming of the strands, and you can get complementary base pairing reforming where appropriate. So let's have a look at the technique. So what you do is you could collect DNA from 2 different species, or maybe even 3 different species if you wanted to do kind of a comparison, and you extract that DNA and purify it and cut it up into small chunks. What you then do is that you label the DNA using a radioactive label, or you can use a little fluorescence marker and leave DNA from one of the species unlabeled. Okay? You then mix those 2 lots of DNA from the 2 different species together, and you then heat up the DNA sample, the mixed sample, and it will separate into single strands when you heat it up. Okay? You then let it cool again, and every place where the base sequence of DNA is complementary on the different strands, hydrogen bonds will reform due to the specific base pairing. Now, they form what's called a hybrid strand, when the DNA from one species in the cooling process actually reforms with the DNA from the opposite sample. Some samples will reform where both strands, as it reforms, are from the same species. But we're wanting in this study just to look at the hybrid strands, the ones that are made up of 50% of one species, of one of which we labelled, and 50% of the other. Okay? Now, what we can say is that the more bases that hybridise, the more alike the DNA is. So basically, when we get two strands reforming together, forming hybrid DNA, the more complementary base pairing we get, the more alike the DNA is. Now, this is the strange bit. What you then do to get an actual value, a number that you can compare, you then reheat these hybrid strands to separate the strands again. And this is the really important bit, because this is the bit you'd apply to the exam question. When the DNA is more similar, more energy is needed to break the hydrogen bonds, and it's because there are more of them between more complementary base pairs, and as a result, a higher temperature is required to separate the strands. Now, this might make more sense if we actually look at an example. So, if we look at this as an example, here we can see, let me just change colour of pens. So here we can see a section of DNA from a chimp, a section from a human, and a section from a mouse. Now, what we would do is, to start with, we would take the chimp section and the human section, and we would, let's say for instance, we'll label the human section so that stands out. We'd heat those strands up together and then cool them down, and within that mixture, as they cool down, we would get some hybrid strands formed. So this bit here would be a hybrid strand, and you can see wherever on the hybrid strand that the base sequence is complementary, where it can form a complementary base pair, we can see we get a hydrogen bond forming. So we've got, these are all our hydrogen. There's a hydrogen bond there and there and there and there and there and there. Okay, so we can see all of those hydrogen bonds formed in this hybrid strand. We would then reheat that strand there and find that actually it took a temperature of 30 degrees C to get that strand to separate. So more energy was required to separate out that strand. We'd then make a comparison and do the same thing again, but this time we'd mix the mouse strand with the human strand and form some hybrid strands. So this would be our hybrid again. Now this time you can see just by looking at it, we only literally got 1, 2, 3. Oh, and we should have another one at the end there. Let me just add that one in. C and G, we'd have a complementary one there. So we only just got these four hydrogen bonds forming, whereas relatively speaking on this side we've got 1, 2, 3, 4, 5, 6, 7. Okay, so when this strand is then reheated, it takes a lower temperature to separate out those two strands, i.e. less energy is required to break those bonds. And so we can make kind of a relative comparison and go, well, the human and mouse DNA must be less similar in terms of its DNA base sequence than the human and chimp DNA base sequence, because it took less energy to break those hydrogen bonds because there was less complementary base pairs. Now, you wouldn't be required to describe any of that technique, just to interpret the actual result. So just to sort of summarise it, we'd say when hybrid DNA strands form, the more similar their base sequence must be, because the hydrogen bonds form between the complementary base pairs. Hence then when you try to separate it out, you need more heat energy to separate the bonds, and so they separate at a higher temperature. So basically the more closely related two organisms are, the higher the temperature you'll need to separate out the hybrid strand. So if we go back and we have one final syllabus check, we should be okay with these top ones because we looked at these right from the start, and we've been through in quite a lot of detail looking at the amino acid sequence of the proteins encoded by the DNA and the mRNA. So we've had a look at that section there. We've also hopefully given you a bit of a chance to look at how you interpret those kind of bits of information in exam questions. What we do now is, hopefully now that you've got this knowledge here, next lesson we're going to be able to kind of apply this to lots of different exam questions and see if you can actually, kind of apply what you've learnt in this. So the best thing you can do is, if you feel like you're confident with it and you've annotated around your PowerPoint and you feel okay, then come ready for the lesson. If you feel like you want to do more reading, then have a go at looking back in the new textbook, looking at pages 249, okay, 249 through to 252, just to sort of consolidate what we've said, and then you can get some extra help in lesson next week. Okay, thanks for watching the video. Hopefully by the time I do the next one, I'll have got myself a good stylus so my writing won't be quite so messy, and I hope it's been useful for you. Okay, bye!